Good morning, everybody. This is the second day of the 2 days webinar that were organized by the NEURON on behalf of this partner in collaboration with EBRA. Focus on the presentations of the resources that European biomedical research infrastructures can offer for researchers in neurosciences. Today we have the presentations of the resources that EBRAINS and INFRAFRONTIER can offer, and I want to thank you, especially our presenters, our panelists, France Nivelle and Lluis Montoliu, that are representing those infrastructures. It's also as panelists Hela Lichtenberg, in representation of the coordinations of the NEURON from the German Neurosciences Center. Unfortunately, the coordinator of the NEURON cannot attend today, but she did the introduction of these webinars on the first day of the meetings. So I will present you an overview of the agenda of today's meeting, and we will pass directly to the first presentation. We will collect the feedback of the participants and also the questions and answers in the end of the webinar. So first of all, we welcome an introduction. Afterwards, France Nivelle will present the EBRAINS resources, as I mentioned before, followed by Lluis Montoliu, representing INFRAFRONTIER, that will present us the resources that are available. They will be available in the end of the meeting for your questions. Try to answer them. Thank you very much, all of you. So I will present briefly France Nivelle, our first speaker. She's Chief Communications and Content Officer at EBRAINS, the Human Brain Project. She's involved in developing and implementing a global communication and strategy, but innovative services are developed by the Human Brain Project teams. And she will involve broad outreach and dissemination of the research outcomes of the project of EBRAINS. Previously, she worked also as Director of Communications in several other holding groups and enterprises. Thank you very much, France Nivelle. The floor is yours.
Thank you very much for the introduction. I will now share my slides with you. And thank you for this introduction. I hope you can see the slides in the right way. Okay, so perfect, thank you. So good morning, everybody. I'm very happy to be here today to tell you a few words about EBRAINS. And I would like to thank NEURON also for organizing this type of conversations, because I think they are very useful, a very useful way to share, what is available to researchers to help progress science, and especially brain science and brain research. EBRAINS, what I would like to do with you today is, first of all, to give you a little bit of background around EBRAINS, but more importantly, to tell you how EBRAINS can help you researchers in your research efforts, with some key research projects you would be involved in, and how together there are possibilities for us to cooperate and to make sure that we enable brain research breakthroughs and advances, which is the ambition of EBRAINS. Compared to Elixir that you had a presentation around about on Tuesday, and to INFRAFRONTIER today, EBRAINS is a very young research infrastructure. It is relatively new. However, it's not because it's young that it doesn't build on a very strong heritage. And actually, EBRAINS is a research infrastructure that builds on and capitalizes on the work that the Human Brain Project has been delivering for the last 9 years. I'm sure many of you have heard of the Human Brain Project. Just as a reminder, this was launched by the European Commission as a flagship project at the time. This was a 1 billion euro project, at least that was the plan. It ended up being a 600 million euro project. The point was very much to bring together different disciplines, going from neuroscientists to IT specialists, engineers, physicists, mathematicians, bringing multidisciplinary teams together in order to help decode the brain. That was the ambition in the beginning. More than 130 academic universities across Europe were involved in the HBP and really focused on different aspects of brain research, working in this translational way on brain science. And this science has actually fed the research infrastructure of EBRAINS, and the infrastructure itself was really very much co-developed by researchers. And the time has come, in a sense, to at the end of the HBP, which is planned for 23, to see what the next steps are going to be, so that the project doesn't end, and finish forever, but that we continue sharing the work that has been done with the scientific community at large through the research infrastructure. And actually, the idea is very much for EBRAINS to offer state-of-the-art brain data and atlases, and I'll come back to all this in a few minutes. But also simulation and modeling tools, and also access to supercomputing, to computing resources, but also to supercomputing resources when they are needed. So important to remember that EBRAINS, although it is very young, does not come out of the blue, and it is the result of the work of 10 years of efforts of the Human Brain Project, more than 500 researchers from different disciplines involved, more than 2,000 scientific publications, and as I was saying, close to 130 institutions working on the project. Now, what are the focus areas of eBRAINS? As I was saying, eBRAINS is fully dedicated to brain science and brain research, and ambitions to enable research advances and progress in 3 areas that are obviously linked to one another. The first one is obviously neuroscience, which is, like, say, the core of the research activity, but also bringing, fundamental research into more applied research in brain medicine. As you know, there is a huge unmet need in brain medicine and in brain disorder treatment, and this is really something that eBRAINS ambitions to contribute to. But also brain-inspired technologies, and I will say a few words about that in a few seconds. By understanding the brain, how can we potentially mimic the functioning of the brain in new technologies? What is the advantage of doing so? But at the same time, we're talking about neurotechnology, which is also meant to enable new treatment in some specific pathologies. If we look at what eBRAINS brings to the scientific community, let me just go through, very, very briefly, the portfolio of services that eBRAINS is bringing. The first one is what we call data and knowledge. This is a very important aspect of EBRAINS, which gathers enormous amounts of data sets, of brain data sets in its database, but that also welcomes databases coming from different sources, from researchers, for example, who can share their data through the platform. So it's very much an idea of finding data, but also sharing data and storing data. The platform gives access to, is also a kind of hub that gives access to other databases across the constellation of databases that we have in brain research. The point is very much to make all this data interoperable, using, of course, as you heard with Elixir, the FAIR principles, and making sure that the data that is gathered is data that is reliable, that is reproducible, and that is curated in the best possible ways. The second aspect and the second portfolio of services are atlases. And by this, we mean animal rat atlases, brain atlases, but also human brain atlases. And the platform really gives, offers a very nice, useful tool for researchers in the form of a 3D viewer of a brain atlas, of a full brain atlas at multi-scale, and you can visualize the various elements of the brain in a 3D viewer capacity, giving you very clear details. But at the same time, these atlases are not like standalone. They can be combined and with the data that a researcher would bring, for example, to allow comparisons and further analyses. The simulation portfolio, the simulation tool portfolio is also extremely important. There are many tools available for simulation. Again, here, targeting multi-scale approaches, tools that focus on specific brain areas, but also tools like the whole brain simulation tool that allows to really simulate things at whole brain level. Which is a very sophisticated thing to do, a very sophisticated approach, but at the same time, this is enabled through EBRAINS. In terms of brain-inspired technologies, as I was saying a few minutes ago, the idea is very much to leverage our knowledge of the brain and the computational capabilities of the models in order to build systems and networks that reproduce the brain networks, the human brain networks, and allow to both develop new types of technologies, like, for example, going into neuromorphic computing, but at the same time, to give back to neuroscience and to learn back from those models to fill the knowledge on neuroscience per se. We also have at our disposal, at your disposal, a medical data analytics platform, which is called the MIP, which is, if you want, our first step into clinical data analysis and gathering. It is a federated platform that for the moment connects more than 30 hospitals in order to analyze and collect brain clinical data and analyze this data, big volumes of data, in order to identify potential patterns, but also potential sources of new treatment. Now, the very specific things of all those services is that they are all interoperable. As you know, today, there are a lot of different tools available for brain researchers that are actually a little bit like isolated, a constellation of isolated tools, which are very powerful and very good, but that don't necessarily talk to one another. What we want to do in EBRAINS is make sure that there is a continuum of services that can really work together. And this is what we express here through this slide. You know, if you use the full-fledged EBRAINS workflow, you really start with data, which can be data that is already available in the EBRAINS data storing platform, but that can also be new data brought by each researcher. This data goes through a curation process and is then shared into what we call the knowledge graph. That can then be compared with atlases of different kinds, then can be integrated into modeling and simulation workflows, the different simulation tools that I was talking about, and if needed, can also get analyzed through a supercomputer access that is granted through the platform. So as you see, the idea is very much to allow researchers to go from data to very sophisticated analytic models, all this in an integrated way. And that's the value, the extra added value that is brought by EBRAINS. The services that we are offering to the communities can be of different levels. We on the left-hand side, you see that we can support researchers in their data management or in their modeling approaches by just allowing them to share their data, to share their models into the platform, allowing them to link their publications, for example, to the data. To publish live papers if they need to do this. I think this is something that all of you are definitely quite involved in, and this is, you know, a key requirement for all researchers. So this is, let's say, the first level of support services that we can offer, and we welcome anybody. This is an open platform. We welcome any researcher to do this. There is a very simple and efficient way to do this. Now, the next level is really to work together with you, researchers, in working with you in trying to understand how the various tools that we have at our disposal can help you answer a specific scientific question that you're working on. This is really like working together, making sure that we can see with you what solutions can be built in order for you to progress your research faster and in a more efficient way without having to reinvent the wheel everywhere, because this is also where science is actually wasting an awful lot of efficient time. It's because there is a tendency to reinvent things all the time. EBRAINS allows and enables people not to waste that precious time, but to concentrate on, you know, going to the next step, going to the next level faster. And the third level of services is very much going into the idea of developing new customized solutions together. Very like it's like a specific project then working on developing new tools, new ways of working, new models, new mathematical models, or new ways of looking at different areas of research. And just to give you a few examples, the first one is the example that I was referring to. This is, let's say, for for the supporting aspect of EBRAINS, where EBRAINS allows scientists to share data, models, and software that they have developed in a very simple way, in a sustainable way, because the storage is guaranteed, the security is guaranteed, and allowing also for researchers to publish papers and in different journals, linking their publications to the data in EBRAINS. So making this easily available and easily shareable with the community. Examples of more, let's say, sophisticated solutions that were already developed with EBRAINS by different projects. If you look at the right-hand side, we work together with a team of scientists in Marseille, at the University of Marseille. The name of the expert is Victor Gersa, who was the project manager, on helping using EBRAINS tools to develop a very sophisticated model, allowing to detect through a whole brain simulation mathematical model, and comparing that with images and images data from a patient, comparing both and predicting more specifically where the epileptogenetic zone of an epilepsy patient would be located in a brain, in an epileptic brain. A lot of patients in epilepsy who need to undergo surgery are still facing a lot of issues, and epilepsy surgery is still a very complex task for the brain surgeons. And this tool would actually add to the various portfolio of tools that a surgeon has at his or her disposal to clearly identify, to better identify the zone in question, and to make more informed decisions before going into surgery. This is a system which is currently being tested in a clinical trial, and we hope to have the results very soon. So just to show you an example of one specific project. The other project on the left-hand side is aiming at trying to better understand the mechanisms of consciousness of a brain. And for this, modeling techniques and simulation techniques were used in order to understand the underlying mechanism of consciousness in patients in awake conditions, but also in sleep conditions and in coma conditions. And the modeling allows to better identify those mechanisms, leading hopefully at some point, when the research continues, to more personalized treatment for coma patients. Once we understand the underlying mechanism better, it might help personalize the treatment for specific patients. And last but not least, I wanted to give you an example also of a project involving brain-inspired technology. By mimicking the functioning of the brain with eye and touch sensors, some research teams developed this robot that is really using, brain inspired behaviors, and teaching this robot in the same way as teaching a brain. We can see that the robot, the sensors, the eye sensors and the touch sensors are reacting to their environment, and are actually using the brain networks for this, which compared to other, let's say, more standard robots, consumes much less electricity. So the robot really functions like a brain. It can be trained like a brain, much faster than a normal robot built on algorithms. Now, I won't go into more details about all these projects. You're of course welcome to get more information, and there is plenty of information on our websites on this, and I can put you in contact with the various scientists if you need to know more. But I just wanted here to illustrate the various ways that people can use eBRAINS. So we can use eBRAINS for supporting the research. We can use eBRAINS to see how a specific question can be answered using eBRAINS tools, but eBRAINS can also be part of a project to develop a customized solution in a specific way. And this is also why eBRAINS today is involved in several Horizon Europe projects and European Innovation Council projects. We are actually supporting several researchers in pursuing their research, in accelerating their research using the eBRAINS tools. And this is what we would like to continue doing more and more in the future. Now, just a few words about the way we are organized. So eBRAINS is a distributed research infrastructure, like many research infrastructures, meaning there is a central hub, and the central hub is based in Brussels. But most of the services that are provided through the platform are services that are provided by best-in-class academic centers across Europe. These are what we call the nodes, okay? Now, of course, it's very important to make sure that all those nodes work together, and this is why the central hub is there, in order to ensure coordination, alignment, and integration of the various services that are offered on the platform. Just a few words to finish. I would like just to make sure that, on top of the services that we are offering, we have several values and a very strong commitment to key research elements. First of all, open science. eBRAINS is an open research infrastructure. It is available to all researchers across Europe, and this is a commitment for us. Open science is a key element. The sharing of knowledge is a value that we are absolutely supporting. The FAIR principles that you've heard a lot about, I think, in the Elixir presentation. So very, very important. And because we're talking about brain, brain data, brain simulation, analysis, mathematical models, responsible research innovation is a key milestone and a key cornerstone of our approach. This existed already in the HBP, the Human Brain Project, in which ethics had a very important place, and this will continue with eBRAINS. This is a very important element that we want to pursue, and that we believe in, and that we are committed to. Three other elements that are linked together. We do believe in co-designing. We do believe in cooperation. So eBRAINS is not there once and for all. It will continue to evolve. It will continue to build services, tools, software, together with the research community. We also hope to be a platform that brings the research community together, so that exchanges can happen and the sharing of knowledge can be leveraged. Collaboration also means that EBRAINS does not want to work on its own necessarily, okay? So collaboration with other research infrastructures, collaboration with, as part of consortia, is very much something that we look forward to, and this is really something that we're going to build over the years. This is extremely important for us. We think that everybody can contribute, and that if everybody brings their expertise together, research can advance faster. And last but not least, impact is something that we look forward to. We would like to be involved in very impactful scientific projects in order to make sure that we can contribute and help researchers achieve their goal, and making brain research advance, progress, not just for the sake of research, but also very much hoping that patients and society can benefit from all these efforts, and that things move forward thanks in part to the eBRAINS tools. Now, we are also very committed to excellence. As you know, because we're part of this presentation, we were very recently added, well, not that recently anymore, but last July, we were added to the S3 roadmap. And as you know, S3 is a very nice recognition of the work that we are doing and building and pursuing. At the same time, the S3 roadmap means requirements in terms of cutting edge and excellent science. So that's all I needed to say today. You can find us, on ebrains.eu. If you want to know more about eBRAINS, there is, a brochure called Discover eBRAINS. What I would suggest is that, don't hesitate to get in touch if you have questions. We would be very happy to answer. If you want to explore the website, the portal, it's at your disposal. And please don't hesitate to register as an eBRAINS user. And we can really help you find your way through the platform, through the infrastructure, and see how it can best enable you to progress faster, efficiently with your research projects and efforts. So thank you very much. I'm looking forward to your questions.
Thanks a lot for your comprehensive presentation. We have received several questions. As we have to initiate again, I would like to ask the attendants to write again the questions, and we will try to answer in the end. Sorry for this inconvenience. So I will present the next speaker. Is Lluis Montoliu, which is a senior research scientist in the field of biomedical science for the study of human rare diseases, with a long research trajectory at the National Center for Biotechnology from the Spanish Scientific Research Council in Madrid. He's also the director of the Spanish node of the European Mouse Mutant Archive, on representation of the European infrastructures INFRAFRONTIER. He's as well honorary professor at the University Complutense of Madrid and representative of the Spanish Rare Disease Initiative, TIBER. Thank you very much, Lluis Montoliu, and the floor is yours.
Thank you. Good morning, all. Thank you, France. It was a fantastic presentation. It's always nice to learn about other infrastructures and the ways we can interact. Hopefully, it will be the same with INFRAFRONTIER. So I will start sharing my screen, and you should be able to see the slides. The slides, you will have them available. I have already transferred to Christina, so you can have them and you can ask any question to me directly or later on. Well, this is yet another ESFRI. This is another European Science Forum for Research Infrastructure that we're talking about. This is in the field of the life sciences, and we have been operating over the last 15 years. So INFRAFRONTIER started around 2007, and it has been this infrastructure where you can generate, where you can analyze, where you can archive, and where you can share animal models for a number of human diseases, including, of course, neurodegenerative diseases and those affecting the CNS that I believe are the ones that are interesting for you. So we have a one-shop system. This is also a multimodal infrastructure with plenty of different countries and institutions involved, but this is transparent to all users and customers. Basically, you are all welcome to enter and access through our infrafrontier.eu. This is our one web page where you can have and find all our facilities and services. As I said, the motto, our objective, our aim is to provide unique scientific resources, services, and expertise to advance the understanding and treatment of human diseases using rodent models. Originally, it was only mice, and in the last years, we have added rat models. So we're talking about rodent, and we refer mostly about mice, but also rats. As I said, this is an ESFRI infrastructure, and it was put together, was coming from previous exploratory initiatives, and we launched the INFRAFRONTIER in 2007. What do we do in INFRAFRONTIER? So we have access to a number of different resources and analytical platforms. And we basically also, we are committed very much with, as Franz was saying, for EBRAINS, with open science, for sharing knowledge and sharing data. So what we do is we develop mouse models for you and rat models for you. We also do systemic phenotyping. As you will see, there's a number of mouse clinics distributed across Europe that are included in INFRAFRONTIER. Of course, we are archiving and distributing these models, and this is also a very important contribution to the animal welfare and the animal experimentation regulation, because we are contributing to the reduction on the use of animals across Europe. We also provide training, and there's a number of courses that we are constantly offering, and we are providing with this the required expertise to either generate, to phenotype, or to do whatever it is required to do with your favorite mouse or rat model. We also do a number of activities related to scientific outreach to disseminate our activities beyond the scientific community. As you know, animal experimentation is always a sensitive issue, and we also like to contribute to this arena by just bringing and sharing the benefits of using the animal models into the understanding and the developing of therapies for treating a number of human diseases, including neuro-related disease. All this data, and this is something that I wanted to highlight, all this data is shared and is available. So as I indicated, fully committed with open science. We're promoting responsible and reproducible research, and therefore, regarding animal welfare, we are fully committed with the triage, replacement, reduction, and refinement. And you will see at the end of my presentation some figures and some examples that I think best illustrate our commitment in this area. And in principle, our main objective is to minimize the duplication of animal experiments by providing centralized, high quality, standardized resources. This is a slide that is summarizing the three main activities that we are offering to interested researchers. Either we are developing the models at the beginning only transgenics, later on transgenics and knockouts, and nowadays, as it couldn't be otherwise, we are providing genome edited models using CRISPR and using other genome editing tools. So once the models are being prepared, are being generated, we are offering the systemic phenotyping through the mouse clinics, and the models that are generated or are deposited directly by the researchers can be archived and can be later distributed. So we are taking from you all the hassle of distributing your favorite model. Perhaps you have published a great mouse model of a given neurodegenerative disease, and there is a number of colleagues that are interested to have your mice, but you don't have the time to be organizing all this distribution. The best way of proceeding is depositing this model into this public repository, into INFRAFRONTIER, the EMMA, which is the origin of INFRAFRONTIER. EMMA stands for the European Mouse Mutant Archive. And thereafter, we will take care, and you will be relieved, and you don't have to worry about distributing these mouse models. As I said, this is a distributed research infrastructure. We have one phase to the customer, and then we have all the therapeutic training, consulting, and as you will see, transnational access calls. So these are where we are and who we are. So we are 29 partners, 27 of them are in Europe, plus Toronto Center for Phenogenomics in Canada, and the University of Tel Aviv in Israel, altogether 29 partners in 15 countries. And you will see that we are pretty much covering most of Europe, and not in all nodes, all activities are offered. So the collection of different nodes, in general, they are all offering generation, phenotyping, and archiving, but not all nodes are offering the three activities, as you will see. As it has been happening with other S3 infrastructures, they have been evolving, and most of them have been converting into ERICs. INFRAFRONTIER is not yet there. We have an intermediate situation. INFRAFRONTIER is based in Germany, in Munich, and due to some peculiarities of the German legislation, we could not organize an ERIC, and they decided to organize a GmbH. This is a private non-for-profit company that was originally formed by these 6 members that you have in the slide, including University of Oulu, the Karolinska in Sweden, the CNRS in France, the IMG in Czech Republic, the Helmholtz in Germany, and the Alexander Fleming Institute in Greece. And this is now being enlarged, and we are now in a transition to become a true ERIC. And once we will be ERIC, the remaining countries that we could not be engaged into the GmbH, we will be joining, and we will be reestablishing this situation. This GmbH was launched in 2013, and we hope that this year, perhaps next year, we will have already the proper ERIC established. The origins of INFRAFRONTIER was the European Mouse Mutant Archive. This is a project that was funded by the European Commission since the early 90s. And the EMMA, it's a subgroup of nodes inside INFRAFRONTIER. It's formed by 16 EMMA centers in 13 European countries, and we also share a common web page, which is INFRAFRONTIER. We have more than 7,000 mouse lines, almost 8,000, as you will see. And basically, what we're offering, these lines are high quality health control, so they all have the European expected standards, and they all have a defined genotype and defined genetic background. And we're shipping this globally to anyone interested, not only in Europe, but also beyond US, etc. This is the list of the 16 nodes that are forming the EMMA. EMMA, again, is a part of INFRAFRONTIER, and EMMA, the objective of EMMA is to archive, to keep, to store, and to further distribute the mouse models to anyone that is interested. And this is, for instance, our role. In Madrid, we are contributing for the generation of new mouse models and for the archiving of the mouse models. For instance, in Spain, we don't have a mouse clinic. We are not contributing to systemic phenotyping, and this is a role that is taken by other nodes. This is just to illustrate that we have been serving more than 700 unique destinations across the world, as you will see, most are around Europe, in the southeast and far east in Asia, and in Australia, some countries in South America, and also in South Africa, and a number of institutions in the United States. Well, this is the work that is being offered from the nodes that have the mouse clinics. What we mean by systemic phenotyping, it is a pathway that the mice or the rats, the different rodent models undergo. This can be either rodent models that have been already deposited in the EMMA, or they can be new models that you have developed your own in your institute, and you want us to do the systemic phenotyping for you. Basically, there will be a chain of events, and there will be different activities being taken care, such as neurology, behavior at the beginning, blood chemistry, dysmorphology, energy, metabolism, cardiology, hearing and vision, respiratory and immunology, further organ systems and disease areas, and eventually, last but not last, histology and pathology. We have been working in reducing the number of animals that are required for entering this channel, and this is the cohort of animals that normally is organized. This is actually covering more than 500 parameters, and basically it's meant to obtain the maximum information from the minimum number of animals, and this has already produced many interesting results. A couple of examples from the oldest mouse clinics we have in Europe. This is the clinic in Munich. This is the German mouse clinic. You have the website. It is part of INFRAFRONTIER, and this is a place where you can send or you can tell them to reconstitute or revitalize a previous animal model that is deposited in INFRAFRONTIER for phenotyping. The second also, it's almost similar in age. This is the French mouse clinic, the Institut Clinique de la Souris in Strasbourg, where they also offering equivalent or similar services, and they also belong to INFRAFRONTIER. As I said, the impact of INFRAFRONTIER can be measured in different ways. This is an account of the different publications that are using the models that we have been providing over 2,600 publications, including, as you see, a number of very high quality publications in the groups of Cell and Nature that have been emerging over the past years through the use of either services or mouse models deposited. INFRAFRONTIER is in communication and in a very close collaboration with the International Mouse Phenotyping Consortium, the IMPC. The IMPC extends beyond Europe, and as you see, there are members located all around the world. And the aim of the IMPC is the comprehensive catalog of gene function. So basically to create knockout animal models for every one of these 20,000 protein coding genes. Why is the connection between INFRAFRONTIER and IMPC? Because the systemic phenotyping is done at the INFRAFRONTIER mouse clinics, and because the mouse models that are generated through IMPC, they are also deposited into the EMMA, into the INFRAFRONTIER, and you can have access. For instance, we are distributing more than 350 shipments with IMPC mouse lines all over the world. This is the website that where you can get into the IMPC data. It's a very simple and very useful, I believe, website where you can type in your favorite gene, your favorite disease, your favorite mouse model, and it will tell you whether the mouse has been created or not, whether it is a planet, and whether, if it's been created, whether it's been phenotyped or not. And if it's been phenotyped, it will give you full access to all the phenotyping data. So this is almost 7,824 knockout genes phenotyped. You can always check what is the latest information available. The latest release of IMPC data is from October last year. And as you see, it's almost 8,000 genes that have been already knocked out, and it's more than 8,000 mutant lines. And this is because for some genes, there are different mutant lines that have been generated, and more than these 8,400 mouse lines have been phenotyped, and you have all the phenotypic data available. What are the different areas where we are focusing our interest? These are three, but as you will see, there are more areas. We have been developing close collaborations with the rare disease community, of course, with cancer, and also with the COVID-19 due to the previous pandemic. And you might wonder whether INFRAFRONTIER and EMBA have any resources that might be of interest for you regarding neurodegenerative and regarding CNS disease. Well, this is very easy to find out, because if you go directly to the INFRAFRONTIER website, at the top right, you have this panel where you can search for the available mouse strains. But if you don't know where or which mouse strains do we have, you can access through human diseases. And if you click on human diseases, you will be brought into this dialog web page, in which you can see that there are different groups of disease models. For instance, nervous system. And if you click on nervous system, there will be a deployed menu with many different diseases that are represented. This is just an example, for instance, for multiple sclerosis. If I select multiple sclerosis, it happens that in our repository we have these three models. And if we click on each of these three models, we will see where are they, and what are the peculiarities, what are the characteristics, and what are the papers that have been associated with these different models, and whether they are available or not. If you see this green dot, it means that they are fully available, and they can be really requested right away. There is also another way we have collaborated over the years with the scientific community, with the help of the European Commission, and this is the so-called transnational access calls. And this is providing accesses either to generating new mouse models, or for phenotyping existing mouse models or rat models, also for generating new rat models. These are provided for free, and the only request that we are kindly taking into consideration with our customers, with our researchers, is that these animals, they need to be deposited into the INFRAFRONTIER, and they need to be made available to the rest of the scientific community. And this is, for instance, one of the last transnational access calls that we have launched last year, was regarding the offering of our mouse models for investigating vaccine candidates for COVID-19, or for investigating antiviral agents for the coronavirus. So the researchers, they didn't have to bother about creating or having the mice in their own institutions. They could send their products, their potential drugs, or their vaccine candidates. They could send into the INFRAFRONTIER nodes, and we were doing all these different experiments. Let me tell you, in the last minutes that I have, what do we do at the CNB? This is just an example of one INFRAFRONTIER node. So we are accepting lines that researchers are depositing, and we are accepting requests from lines that we have that are distributed. The depositing is for free, and the only thing we request to the depository is that they have to cover the shipping cost. And we can also accept a grace period up to 2 years. That means you can deposit already your mouse model with us, and then you have those 2 years for publishing your paper. And during these 2 years, the mouse model will not be shown in the database, will not be searchable. Also, if for whatever reason you have a material transfer agreement, it travels along with the mouse. We are not doing anything, we just intermediate. And if your institution decides to have an MTA for anyone interested in your mouse model, we will attach the MTA, and we will make sure that anyone requesting your mouse line will have to fully sign and fully execute the MTA before the mouse line is being traveling. The only requester, the only cost is for requesters, for researchers that are requesting mouse lines, and we are requesting a repository fee, and we are promoting the use of frozen sperm or frozen embryos. As you see, 1,600 plus shipping cost, and if you want to have real mice, if you want to have live mice, we can revitalize the mice for you, but then it will cost double, will cost like 32,000 euros plus shipping costs. We also offering isolator units, and this is of importance because, as you know, sometimes the mouse models, they have a very peculiar health status, and they are not being accepted in some institutions. So we have these isolator units so that we can receive these mouse models, we can obtain the corresponding embryos or sperm, we can cryopreserve them, we can clean them all, and then we can redistribute this with FELASA standard conditions to anyone that is interested. What are the procedures that we are offering at the CNB, but also this will be similar to any one other node in INFRAFRONTIER doing EMMA services. We are cryopreserving embryos, sperm, or oocytes with genotyping, and we are rescuing. We're thawing these mouse embryos, sperm, or oocytes. We're doing in vitro fertilization, we're doing embryo transfer, and we're doing also genotyping and quality control. We have a number of different procedures, and this has been changing a lot over the years for importing and exporting mouse lines. Before, it was pretty much only distributing and receiving mouse embryos or sperm in liquid nitrogen, but now we can also receive embryos that are refrigerated and sperm inside epididymus that are also refrigerated. And this has been a great contribution because that has been speeding up the process and also allowing many of our colleagues to be able to send their mouse model to us. Because, for instance, if there is a given mouse model that is awfully contaminated with some pathogens, it will be very difficult to be accepted, even in the presence of isolators. But instead of traveling with these very potentially dangerous mice, what we can accept is, for instance, epididymus, and these epididymus, from them we can obtain the corresponding sperm, and we can cryopreserve the sperm. This is the system we're using. This is developed in Japan at the University of Kumamoto at the CART Institute, and this is the system we are using for refrigerated. Refrigerated means between 4 and 8 plus degrees Celsius, especially for epididymus. We are storing and we are maintaining embryos and sperm totally immersed in liquid nitrogen tanks. This is different from what you probably are used to see for cells. You know that in cells you have some liquid nitrogen at the bottom and the rest is in the vapor. Here, what we try to with these automatic refilling containers, tanks, we have them totally immersed, and also we have a backup samples stored at a second independent location. This is mandatory for each EMMA node. We are not only having different copies of every mouse line at different places in one of these tanks, but also we have in different tanks. For instance, this is the campus where we are located, and we have a split archive between our neighboring institute, the CBM Severo Ochoa, which is 600 meters away. So if anything happens in our institute, there is always a backup possibility to rescue the line. So this is how we store the sperms and the embryos. These are the straws, and they are also grouped in these goblets that are immersed in liquid nitrogen. There has been a number of technical advances. The cryopreservation in mice and nowadays in rats has changed a lot, because there have been a number of innovations, mostly coming from CART. CART is the Center for Animal Research and Development at the University of Kumamoto in Japan. And they also provided all these different and new techniques that have possibilitated to do all these techniques with an incredible high efficiency. So in summary, what we do, if we receive males or we receive epididymis, we obtain sperm. The sperm can be frozen and can be stored cryopreserved. This sperm can be thawed, and then we can use this sperm either fresh or upon thawing with an in vitro fertilization with oocytes that we can obtain from females, or we can have them also cryopreserved before. We have the embryos, we quality control these embryos. These embryos can be frozen or can be thawed and eventually can be transferred to a pseudo-pregnant female for rescuing the corresponding mouse line. The procedures are now include in our note the generation of genome-edited CRISPR mice. We are using the electroporation of mouse embryos, and we are offering gene disruptions, gene deletions, and gene substitutions. And we can offer this because INFRAFRONTIER is one of the few institutions in Europe that we have worked out and finalized a CRISPR licensing agreements in Europe with Caribou and Earth Genomics. As you know, the CRISPR owners are different in Europe and States. In Europe, the rights belong to Caribou and ERS Genomics, and in the States, they belong to the Broad Institute. We have CRISPR license agreements with both, so it means that we can generate and we can distribute clean and absolutely legalized mouse models to any one of you, even if they have been produced using these CRISPR genome editing tools. How many lines do we have in one node as compared to the rest? Well, as you can see, here we have at the CNB more than 500 lines out of these 7,000 that we have. This is about 7% of all the lines. And what is normally constant across the different nodes, it's about 1/3 of the lines that are being requested, and 2/3 are normally not requested, and perhaps they will be requested in the future. If we take into account how many times the different lines are requested, as you could see, there are many lines that are only requested once, but as you can see, we have some bestsellers. We have some of the mouse lines that are being requested more than 30 times because they are useful, because they are CRE lines, or because there is a gene that is very popular. I want to finish by just providing you some hints about our contributions to animal welfare. I told you at the beginning that working with animals, it's always sensitive, particularly in Europe, and there is always pressure from the groups that are against animal experimentations. They want to reduce this as much as possible. And so we are providing examples, how are we contributing to the 3 Rs and how are we contributing to animal welfare. Let's say replacement. So we can quantify the number of animals that we save because the animals are not alive, they are frozen. So if we have more than 7,000 mouse lines cryopreserved, and we assume, and this is a fair assumption, that we need about 50 mice per year to maintain a given mouse colony, if you make the multiplications, it's up to 350,000 mice that would be there that are not there. So these are mice that we have been saving. So these are animals that, because we maintain all these lines frozen, they have not been generated. And this is, I think it's a remarkable figure to take into account. Also, reduction. How do we reduce the number of animals in all our techniques? Before, for cryopreserving one mouse lines, we required between around 10 males and around 50 to 60 females during 5 to 6 weeks. And that was a very tedious and very cumbersome process. So it took us 1.5 months to cryopreserve one mouse line. But now, because of the innovation and the new methods, we can use only about 2 to 5 males and 2 to 5 females, and we can do everything in 1 day. So we go from 60 to 70 mice to 4 to 10 mice, and from 6 to 7 weeks to 1 day. I think this is also remarkable. And regarding refinement, before, for the quality control, you want to make sure that once you have cryopreserved a mouse line, you can rescue a given genotype. So you can thaw these embryos and you can obtain exactly the same allele you have cryopreserved. Before, this was done generating pups, and nowadays we have streamlined the process, and now we are genotyping individual blastocysts. This is a sophisticated technique that is allowing us to also save animals. So the animals, they are not brought to term, and basically we can double check whether everything went as it should be. We do the quality control with this blastocyst genotyping. So I'm finishing by just presenting, this is my colleagues at the INFRAFRONTIER node in Spain, in Madrid, at the CNB, Julia, Maria Jesus, Marta, Cristina, and Ines. This is our website where you can find us. And this is also the email address where you can send any request, cryocnb@CNB-CSIC. Thank you, and I'm ready for any question. Thank you.
Thank you very much, Luis. Thanks a lot for this comprehensive presentation. I think it was very interesting, both of the presentations for the researchers in the neuroscience field. So I think that we can begin with the questions and answers session. The first one is addressed to France. Does EBRAINS have a data management policy in place? Is it accessible to users on your website?
And the answer is the data management policy is being updated as we speak, and it will soon be available on the website. So yes, this is a commitment. We want to have a clear policy about this, and it will soon be available to all.
Thank you. Thank you very much. The next question is from Maria Alonso. Thanks for your presentation, Franz. If our institution is a member of the Human Brain Project and EBRAINS, should researchers register individually or as members of the institution?
Researchers need to register individually, but they need to use their academic address. Okay, when they register, and then they will automatically be registered, and we will know which institution they come from. Okay, but registration is individual and using the institution address.
The next one is, you mentioned 2 medical data analytic platforms of EBRAINS in your slides. Could you expand on this information, please? Is from Sabine Holter.
Yes, and for this, actually, I've got a team working with me. And I think, Martin, if you could unmute and maybe give more explanation on the 2 platforms. Okay, if you can unmute, Martin, please say it out loud because it's in the chat, I think, but
by the end of Human Brain Project, 2 will be available. The MIP is available, well, it's in the chat. They can consult the second service. The IP is not available yet. More information about it can be found at the other address. So you can consult the other address.
Yes. So
everybody can see the information.
Right. The MIP is available. The HIP the health informatics platform is still in development and will be available soon. Okay. And then I will also ask Martin to answer, the last question.
Okay, I can give him permission, maybe. Well, the last question is, how about multi-center validated protocols, such as immunohistochemistry, neuroimaging, analytics? Are they available? In case that you want, I can give permission. So if not, I can read aloud. The answer is yes, data can be linked against a wide range of protocols, such as data generation, data processing, feature extractions, analysis, classification, etc. And he provides the general information on the website and more information about the extendable metadata scheme.
Thank you, Martin.
Thank you. He added something else. Sorry. That he provide the contact email for further questions in case that maybe sure. we can provide all this information maybe also in the website of the NEURON sure. Afterwards.
With pleasure, and don't hesitate. Okay, and this email address is at your disposal, and we'll make sure that you get the most specific answer.
Okay, and any other questions that I think that someone has permission for?
Christina?
Yeah, hello.
While you're looking, may I ask for something?
Yeah, please.
Yeah. So thank you very much. It was an excellent presentation. May I, because Lluis quite well elaborated on the details of accession and the workflows and everything. So may I ask the related question to you? How is the workflow? So you just said each person individually has to register as a user. And how is the particular involvement of EBRAINS in, let's say, a specific project? So, just for an example, in Neurons, we publish annual calls. So a research team finds themselves with a great idea, with a good innovation plan, and they think that some platform services might be of use. So how would they proceed?
Well, that's a very important question. So what we do in that case is we would love for them to contact us, and then we start putting a team of researchers together in order to see what the project is about, what the scientific question is about, and how EBRAINS can contribute to helping this project develop and progress. Okay, so this is really like a pre-proposal kind of discussion that we need to have to see in what ways would EBRAINS contribute to helping the research project. So these are discussions that we have prior to the proposal writing. And then if we see that there is a match and a fit, then we're very happy to enter into a consortium or any kind of agreement with the project leader in order to make sure that we work together on the project.
Okay, so Lluis mentioned the material transfer agreement for the mouse lines, and he also mentioned some sort of embargo for people that want to have protected their results until the publication is out. So do you have anything related, let's say, a confidentiality agreement? Because if people approach you with, let's say, a real stylish, fancy idea, they might not like to discuss that in a really huge round or that distributed among thousands of people. So do we have something available and at hand for confidentiality agreement, material transfer agreement, anything like that?
Yeah, no, absolutely. We've got a confidentiality agreement when discussions are, under embargo and when they are private in terms of IP, etc. So this is absolutely something that they can rely on. Okay, we want to be a trusted partner, so we'll make sure that all those requirements are met and well defined before we enter into conversations. So there's no problem on that front.
Thank you. I didn't thought that there's any problem. I just want to clarify how the workflow might be
and
for
you're absolutely
our
right.
interest.
Yes.
Yeah. Thank you very much.
This is important, what you were highlighting, Hela, because in order to provide good services, the researchers, they need to be comfortable. They need to, I mean, we are all for sharing, but also at the same time, we want to protect our ideas and our inventions. And this is something that all the different infrastructures in one way or another, I think we all taking this into very good account.
Thank you very much.
I wanted to ask Lluis a question. Have you been integrated as partners in any NEURON funded research transnational projects as infrastructures? Because I think that is sometimes a bit a hurdle to integrate, not only to apply for the services, but to integrate as real partners, the infrastructures.
Exactly.
Well, not that I know at the level of Spain or at the level of Europe, but as you can imagine, we are eager and most happy to collaborate with all of you. In this case, you have a clear interest in CNS-related mouse and rat models, and this is what we can offer. And also the systemic phenotyping include some behavioral studies and include some other neuro-related parameters that can be obtained. I'm sure most of you have already the equipments and the methods available, but if not, you can always think about sending this your mice to one of these clinics. And in principle, this can be done through two different pathways for the clinics. Either you set up a collaboration, and this will cost you nothing. Basically, it will be a scientific collaboration. Or if you want to follow your private interest, you will pay the phenotyping fee, and that's it.
Well, in that case, it's a good opportunity for us to clarify here. If an infrastructure is involved as a partner in a project, which is possible, at least some funders in NEURON, we haven't made a recent survey on that. We did that already some 7-8 years ago. The situation might have changed, but at least for us, this is fundable. So if you offer a service in phenotyping, archiving, anything, it can be a budget position in the financial plan of a project, and this is fundable.
Oh, this is interesting. This is very good. This is very good. I mean, for many years, we and other infrastructures, I don't know whether EBRAINS had this phase, but for us, we were receiving some funds from Brussels. But since I believe I think it's more than 6 years, we haven't received funds anymore. So we are now floating on our own, and we are on a sustainable phase. So basically, we are generating the funds that we need to operate the infrastructure together with the national supports. So every state member that has nodes, it is also providing support to the corresponding nodes for this infrastructure. So the idea is either to provide free services to the researchers, and if there is some cost to be transferred to the researcher, this has to be kept to the minimum, only to repository fee or to the real cost fee.
Well, I mean, as far as I understand, the construction of an ERIC is that your, institutional costs, your maintenance and sustainability costs, these are covered by the country membership fees. However, the direct project connected work you are doing in a specific research project in a scientific approach for the next 3-5 years. So that is something that could be funded.
But this is, I told you, INFRAFRONTIER is not yet an ERIC.
Yeah.
So
Right.
we're still operating as a company. We're still operating as a GmbH.
Yeah, but still, I mean, this is not most of yeah, I mean, if you involve a pharmaceutical company or anybody else, or a startup in bioinformatics, that would be, in my understanding, a related situation.
Well, let me explain you. This is different. All what we're offering, this is for academic institutions, for public research. If there is any private company that is interested, they need to. We need to set a new agreement. So they will be different. They are not being offered the same conditions for private as we offer for academic institutions.
Perfect. Thank you for the clarification. And I apologize that I have been misunderstood. My comment was directed to the fact that your current legal status, which is not yet an ERIC, it is still a legal status that is in principle also enabled to receive public funds.
Yes, and this is actually what is happening. The DFG is giving a good amount of money for this INFRAFRONTIER in Germany, for instance.
For instance, okay. So we have clarified that one. Thank you very much.
Thank you very much. So, are there any questions? No? Or if you want to comment anything else, Lluis and Franz?
That we're happy to work with all of you. So just send us your request. We have plenty of mice potentially interesting for you. Just have a look. Sometimes people simply doesn't know that the mice are already been made.
That is the point. That is the point. I think
And
that really needs
this
a
is
bit
my
more
always,
advertisement.
this is my first suggestion whenever I have a colleague coming to us and say, well, we want to make this new animal model. Say, wait a minute. Have you checked whether this animal model has been already generated by someone else? Maybe you can benefit.
Yeah, I think that your last slides were really impressive of the amount of mice that can be saved.
Well, this is important because we have a pressure, as you probably know. In September, the European Parliament passed a resolution in which they were urging the European Commission to finalize animal experimentation as soon as possible. Well, this is the eventual final aim of our legislation, but of course we need to read the complete phrase, and then we have to terminate animal experimentation when it would be scientifically possible, which is not the case yet. And now we're having again another wave of pressure from the groups, from the animalist groups, that they are against use of animals. They claim that alternative methods, they are already available for all experiments. Unfortunately, I wish this would be true. This is not true.
No.
And so we need to make clear that what is the benefit of using these animal models. And also we need to make clear that we take very seriously the recommendations about replacement, reduction, and refinement.
We have received one last question for you, Lluis, from Sabine Holder. Can you expand on the creation of new genome edited mice, especially humanized models?
Well, for the humanized models, depending how you define a humanized model, if you define this by the replacement of a human gene with the corresponding mouse gene, well, this is a specific experiment that is not covered normally in this transnational access, because this is a knock-in and this is still a challenging experiment, which doesn't mean it cannot be ordered, but this is beyond the activities of INFRAFRONTIER. Some nodes, they might be offering this depending on their capacities and etc. So this is what we do. For when we do genome edited mice, we mean inactivation of genes, we mean small deletions, or we mean short deletions. This is what we do.
Okay, thank you very much for the clarification. Thanks a lot, both of you, for this presentation. I think that it's very important to give further dissemination of the services that can be very useful for the researchers, and maybe they are not aware of what this is 16. That's I think that's one of the main purpose of this webinar. So we wanted just to launch some poll questions just regarding the inclusion of the infrastructures on the research projects. I will, if the attendees can give them their answers.
This is only for participants. We are not supposed to.
Yeah, we are
Yeah,
not
sorry.
supposed to vote, unfortunately.
No, it's a kind of feedback for the
No, we cannot
for
cheat the
the
system.
utility of these webinars. I think that they were very useful, and we will try to further disseminate as this will be kept recorded. So because I think that this can be useful not only in the field of neuroscience, because we made this, organize these webinars on the framework of NEURON, but also for other fields. I think that the services that you offer are very important for researchers.
Certainly.
So in general, yeah, they will consider an option, or for sure that they will try to involve research infrastructures in the next projects, research projects. And regarding the feedback of the webinars, we would like to know if they were useful and interesting for the, or if they would like to receive more information of other infrastructures. So far, we presented Elixir, EBRAINS, and INFRAFRONTIER, and also the data management tools that were very, very useful for elaborating the data management plans that now are mandatory. So, yeah, in general, they found them very useful and are interested in future webinars on this sense. So we will try to see our best. We will try to do our best.
100%.
Yeah, thanks a lot, Lluis and Franz, for your attendance, and thanks, Hela, also for the representation.
I would specifically thank the both speakers. That was marvelous presentation, very important for the science community. And we really hope to meet in the future and can repeat that in one way or in the other. And we will strengthen our efforts in order to advertise really more, because that is the future.
Thank you very much, all the attendance also.
Looking forward to hearing from you.
Okay, thank you.
Bye-bye.
Bye.
Bye.